Hey everybody, Miss Chatsy here. Thought we could take a minute to talk about the remainder of material for the unit that we're studying with inheritance. So we spent a lot of time talking about DNA, so now it's finally time to talk about the long-awaited RNA. So just to start out structurally and purposely what RNA is all about. So as you probably picked up by now, DNA is really just sets of genes, so it's code that tells your body or tells the cell what it's going to make. Now RNA is the actual molecule that does the work, so it creates proteins. So DNA is a code that tells a cell which proteins to make, and RNA is the one that's going to be actually assembling the protein. So DNA has the info, but it doesn't really do any work because it never really leaves the nucleus. RNA, however, has the ability to maneuver around the cell and construct a protein. So let's talk about the differences structurally between the two. So RNA, first off, let me just start at the bottom here, is a single helix instead of a double helix. So it's just simply a winding, single-stranded molecule. It depends on the length, just kind of depends on the size of the gene that you're working with. There's going to be some other structural changes. One of them is the sugar. The sugar is replaced with something called ribose. So actually, RNA stands for ribonucleic acid. Nucleic acid. There you are. It still contains a phosphate, so those will be in the same position, and the nitrogen base will be in the same position. So structurally, you'll still have everything made up of nucleotides. But what's different about the nucleotides, besides the sugar, the type of sugar, is this right here. Because of the number of bonds available in the ribose sugar, thymine cannot bind with ribose. So there is a fifth base called uracil. So we pretty much just now replace T with U, and we make a single-stranded molecule that will have sequences of A, G, U, and C, and that particular code will pertain to one particular protein that we're going to make. Okay, so I included a little bit more detail if you want to know some more about the bases. It's not super necessary for you to know the molecular structure of them. Do be aware of the base pairing rules. So this right here, cytosine and guanine, that's the same as it was in DNA, but what does change now is that adenine will now pair with uracil instead of pairing with thymine. So when we're making RNA, pretty much you won't hear thymine or T correlated with RNA whatsoever. We're just going to use uracil. So once we get to some practice problems, I think it'll make a little bit more sense. Okay, so what we're going to be doing here is assembling a protein. It's called this protein synthesis to make a protein. We did quite a bit of this freshman year. I think some of it will seem familiar, but I know it was a long time ago for many of you. So DNA contains the information, but RNA actually is responsible for assembling and building a protein. Oh, by the way, in case you don't remember, proteins are made in the ribosomes of the cell. So when we, or I don't even know if we did it freshman year, but I know in junior high, when you were drawing pictures of cells and drawing the nucleus and the ER and the Golgi bodies, when it came time to draw the ribosomes, probably your teacher had you just do something like this. So you're making dots all over the page. Ribosomes aren't really true organelles. They're not made up of a membrane like most other parts of the cell are. So they're not really included in that respect, but there are many of them in the cell because your cell constantly has to be making product. That's how you're able to do pretty much everything that you are able to do. So we have lots of ribosomes working, synthesizing many different genes. Something else that's different about RNA is that there's actually more than one kind, and some of these are originals and others are things that we've recently discovered. The first one is called mRNA, And the M actually stands for messenger. So sometimes you'll hear it referred to as messenger RNA. It has a particular job in that it just transfers information from one location, the nucleus, to the ribosome. So it doesn't actually make the protein yet, it's just delivering a message. Then we have something called tRNA, and the T stands for transfer. And its job is going to be to actually transfer the building block of a protein in a particular sequence. And those building blocks are called amino acids. We talked a lot about amino acids in evolution because it's one of the ways in which we measure relatedness. How many amino acids do you have in common? How many genes do you have in common? So tRNA's job is to actually kind of start to build that or assemble those amino acids in order. There's something called rRNA, and the little r stands for ribosomal. This one you should never forget on a test because it's basically what ribosomes are made of. So because they're not true organelles, they don't have a membrane around them, and it's, by the way, something that even bacteria have. Ribosomal RNA is made up, it makes the ribosomes, more or less. We'll show you a picture of that a little bit later. There's a relatively new one. It's not that new, but it's kind of new. And we're going to talk mostly about this one in the next unit. And it's called RNAi. The I stands for interference. We've pretty much discovered that not only are there pieces of RNA that help to synthesize and turn genes on and make them, but there's actually pieces of RNA that turn genes off. That silence. We call them gene silencers. And there's a lot of things that can cause, and there's a lot of sources of RNAi. We'll talk about that later. Okay, so included a picture of all three. Here's your single-stranded messenger RNA. This is the cartoon version. Remember that you're going to have your ribose and your phosphate and your ribose and your phosphate and your ribose and your phosphate. And then these, of course, are the bases. So this would be something like A, U, G, and C all along here, single strand. Here's what a ribosome looks like. It's actually made up of two parts. There's the large, what they call a subunit, and then there's the small subunit. And they show you the ribbon structure to show you the ribosomal RNA. And then my favorite one is tRNA because it looks cool. It actually kind of looks like the letter T, a little bit of a lowercase t there. All along this ribbon, you're going to have bases. They've chosen not to draw them because they want to focus on this piece at the bottom. These three bases at the bottom are the most important ones. So we'll get to them in just a bit. I'm going to skip RNAi for now, but there's a little bit more information on it. As I mentioned, we're going to talk about that one a little bit later. Okay, so let's talk about how it is that we make a protein. So there's a little bit of a dogma, a mantra in genetics that we say DNA becomes RNA, which then becomes a protein. So it's basically making a sequence, assembling a bunch of amino acids in order, and then you make that particular protein. There's a two-step process. Step one is called transcription. And transcription is when you take your DNA and you transcribe it into a new message. That's called messenger RNA. You're not actually building the protein yet. We're talking about this step right here, turning DNA into RNA. This happens in the nucleus because it turns out that's where DNA is. So let's say that there's a gene that I need to turn on. Let's say I had dairy for lunch. And I always use this example because it's the first one in my mind, but dairy products often contain the sugar called lactose. In order to digest lactose, a cell would need to produce something called lactase. And lactase is a protein. Particularly, it's an enzyme, but it's a protein. And so when lactose is present, I might actually produce this lactase. So somewhere in my double helix is the gene for lactase, and I would want to turn it on and transcribe it so that I can make it. So there's an enzyme that helps do that. It's called RNA polymerase. It binds to a particular part of the gene and then transcribes that gene. What you're going to make is something complementary to the original DNA double helix. My dog is snoring in the background, sorry if you can hear that. Okay, I'm going to show you lots of pictures and we're going to watch a little video later about this, but in blue here is your original double helix. So what will happen is it looks a lot like replication, where you're going to have an enzyme that opens up the helix. Let's say right here, this is the gene for lactase, and I need to make some lactase. I will assemble in red here, this is the mRNA being built from scratch with complementary base pairing rules. So T would pair with A would pair with U, G would pair with C. Now notice there's T's here, but they're part of the DNA, so that's okay. What we will not have is any T's in red. So this that you're building here is your newly made RNA, or messenger RNA. So what would this look like on a test or a quiz? Well, practice question might be, if I give you a sequence of DNA, can you transcribe it into RNA? So all you got to do is base pairing rules. T would pair with A now would pair with U, and C would pair with G, so that's normal. And then we have T with A with U, C with G, G with C, and A with U. Okay, now this will go on and on and on until it reaches a particular sequence at the end that is codes for stop, and that signals the end of the gene. So every gene has a start and a stop. So, by the way, AUG is always the start codon as well. So let's get to this word codon. When we read DNA, we read it in bases of 3, and each of those 3 is called a codon. So this is a codon, and then this is a codon, and then this is a codon. These codons will correspond to a particular amino acid. So for example, AUG, I would look at this table, and you would never ever have to have this table memorized. It's just something that will always exist for you. And you would search for, what was it? It was AUG on this table of codons. So they all intersect, but you can kind of just scan and look for it. AUG is right down here. That's the start codon. It actually codes for the amino acid called methionine. Now, oftentimes, authors will abbreviate it as MET. So every gene actually starts with that, believe it or not. Our one up top here was CAU. Let me rewrite that. Then I would find CAU on my chart here. It stands for histidine. That's another amino acid. We'll just abbreviate it as HIS. And then the last one here was GCU. I'm just transcribing that from the top. And GCU stands for alanine, so ALA. Now, I did not make a stop codon, but you will note that there are 3 particular codons that actually do code for stop. Now, why are they these 3? That is a mystery that we do not know. We just know that there's a lot of different ways to end a gene. So this would continue on until we'd hit one of these stop codons. So all these guys on here, these funny long names, these are all called amino acids. Amino acids. Okay, so it's not quite over yet. We have a few things left to do. We have to fix the mRNA transcript. This is something special that we did not talk to you about freshman year. We have to modify it. There's a lot of stuff in a gene that doesn't necessarily pertain to the gene. And since we're going to be sending this mRNA out of the cell and out of the nucleus and into the cell, it's going to be interacting with a lot of other chemicals in the cell. So it needs to be protected before it gets to the ribosome. So things that we do is on one end we put a cap, which is made up of a special nucleotide. On the other end we put what's called a tail, which consists of like 3 adenines all put together. What this does is it provides stability. It makes the mRNA not want to break down over time, so therefore it can actually get to its destination. The other very, very important thing that we're going to do is look at what's in this transcript that we have made, and is it all relevant to constructing our gene. So there are 2 pieces that could exist in a transcript, introns and exons, and they often get confused by students because it's not exactly obvious which one's which. Introns are non-coding, meaning that they're not part of the gene. They don't actually help us make the protein, so we actually remove them. So introns get removed and exons get spliced together, meaning you cut out all the introns and you squeeze everything back together in the middle. Exons are named because they are expressed. So gene expression means that I can actually see the gene expressed in my phenotype. It's evidence that the gene has been made. So I included a little detailed picture here of all the different parts of a gene. So here's a sequence of DNA. There's all kinds of stuff, by the way, on a DNA double helix that has nothing to do with genes. And we'll talk about all these in the next unit, but let's just look here. We've got the promoter. So this is where RNA polymerase will bind. By the way, RNA polymerase is just like DNA polymerase, except instead of making a copy of DNA, it makes RNA. Same idea, so a little bond here, and it will transcribe, etc., etc. Some of these things are introns and some of them are exons. This is going to be called our preliminary transcript. We're going to remove the introns. Okay, they get spliced out. They're junk. We kind of recycle their pieces. And then we squeeze everything together. It's got a start codon. It's got a stop. It's got a cap. It's got a tail. This final product here is called a transcript. And this is going to be heading to the ribosome, which is its final destination. Okay, so there's a lot of stuff in your DNA that isn't necessarily part of a gene. It's really just instructions on how to make the gene. So it's a very detailed model of how to turn a gene into something. Okay, almost done. Step 2. Step 1's the longest one. Step 2 is called translation. So back to when we were looking at our central dogma of DNA here, this is this part. Translation is how you turn the RNA into an actual protein. So proteins are made in the ribosomes, so we are officially in the ribosomes, which are all over the cytoplasm that we drew in that little picture there. And we're going to build our amino acid chain. tRNA comes into play here, as well as rRNA, because we're going to be working in the ribosome. So the best way to just kind of show it to you, or explain it to you, is to show it to you. So here's your mRNA, single-stranded. How do I know it's RNA? It's got U's in it. This big giant guy is your ribosome. So your RNA is going to feed through the ribosome kind of like a car wash. And as it does that, you've got these little tRNAs hanging around in the cell. We pick up tRNAs, we make them, we pick them up through our diet as well. That's where some of these amino acids will come from. The tRNAs contain a very special sequence on one end that we call an anticodon. An anticodon will match up with a codon. So for example, AAA is the codon, and then UUU is the anticodon. So they're just opposites. So these will collide, and when the right collisions take place, a bond will occur. When that bond occurs, it deposits and releases the amino acid, and then the tRNA kind of floats away, and then it's going to probably pick up a new amino acid later on. As this is moving through, you pop one on, you pop one off, all the correct bases are matched, and then you start to build a chain. This is called a polypeptide. Peptide is the type of bond that holds the amino acids together, and since there are many, it's a polypeptide. When this peptide gets long enough and it reaches its stop codon, wherever that's going to be, then we would officially identify it as a protein at that point. Okay, so it's easier to see it in motion, so I've got a few videos that I want you to watch in a little bit to help you do that. But in summation here, here's your double helix. Here's transcription, turning DNA into RNA. Notice the U's, so this is your messenger RNA. And translation is all about building the sequence of amino acids. These are the symbols for those amino acids. Okay, more to come later. See you soon.